with our July edition of the Overlife, and I'm Runsi, the founder of Ovarcome. And today I'm joined with Dr. Coleman, and we have the privilege of talking to him about new treatments, drug developments, and clinical trials for ovarian cancer. And Dr. Coleman is not only a global expert in this field, but he's also a great champion of Ovarcome and serves on our advisory board. Thank you so much. So grab a chair and join the conversation for the next 20 minutes or so, as we have so much to discuss. And our apologies, we are a little bit over time.
That's my fault,
Dr. Coleman was overcoming his own schedule. But back to the live, we strongly encourage comments and feedback. And if you have any questions for Dr. Coleman as we go along, please type in the comment section as we watch. So thank you so much, Dr. Coleman, for joining us today, and it's always an honor. Yeah. I have a lot of questions, as I always do, so I'll just delve right in. So my first question is, new research is still suggesting that women with ovarian cancer are not undergoing genetic testing as much as recommended. And can you tell us more about the importance of BRCA testing for ovarian cancer? And how genetic mutation might impact and guide treatment decisions for patients?
Yeah, so there's two, this comes up a lot, and you're right, we're still so surprised that we don't have better traction on this. And we've been talking about this for 25, 30 years now. And but when you think about this understanding BRCA and what its implications are to patients, and families, you learn that the gene itself, or understanding the whether or not the gene is mutated in individual patients or in tumors, has both prognostic and predictive value. And the question you asked actually has both of those components in there. So what do I mean by prognosis? So prognosis is it's a situation where if the factor is present, then that either has a negative or positive impact on expectations for outcomes. It doesn't relate necessarily to a specific drug or product in how a patient will do. So we have lots of prognostic factors that are not related to drug therapy. So what you'll see when we see the outcomes of patients, let's say, that have a specific biomarker that has prognostic but not predictive value, we find that those patients either do better or worse as a group, and that the results of the trials essentially show that the benefit is the same, but it's just better or worse in the prognostic group. So that's prognosis. And people always say, well, what's my prognosis? Well, that's, in general what they're talking about is like, what's the likelihood that they're going to be, have a progressive free event or a response or a survivorship or whatever. Those are helpful. They're informative, but they're not really what you're getting at. And really what you're getting at is, what's the value of knowing that, because we have something to give patients, right? That's predictive. And so BRCA is one of the only, if I can think of across all GYN, it's probably one of the only biomarkers that have both prognostic and predictive value. And the predictive piece of it is why there's so much interest in it. And we talked about all the therapies. You said guide therapy. Well, we would use this to guide therapy if we had something that we would give to patients who had it that we would not give to patients who didn't have the biomarker. That's the predictive component of the biomarker. So it has tremendous importance for us today, and it's becoming even more important because these drugs, the PARP inhibitors, and some of the other drugs that we're combining with the PARP inhibitors, are demonstrating value amongst multiple different cohorts of patients. But the index group of patients, the ones that we know for sure that these drugs have some value, are the patients or the tumors that have the mutation in BRCA.
On PARP, my next question. So as we understand, they are changing the landscape for ovarian cancer treatment, right? Could you share the latest guidelines and results that were presented at the recently hosted ASCO meeting? And you don't have to get into big details, but just what it implies for patients, both for the mutation carriers versus non.
Yeah. So I just I gave kind of a historical update not too long ago at a meeting about the PARP inhibitors, and it's actually an amazing story, because if you go back to where this all began in identifying a potential clinical avenue for the use of these drugs, it was in 2005 that we had two papers that were published in Nature that basically said, hey, listen, if these cells are deficient in BRCA, you give a PARP inhibitor, they're super sensitive. Okay. 4 years later, we had our first publication in the New England Journal of Medicine as a phase 1 trial. And in 2013, we had none of these approved. And now in 2019, we have 3 drugs, hopefully 4, that are approved in 3 recurrence maintenance indications, 2 treatment indications, and 1 primary indication for maintenance. So think about that. In that short period of time, in 14 years, we went from a lab to multiple drug indications. So this year, adding to the further database of all of these potential indications, we had SOLO3 presented, which was a study that was looking at these drugs versus chemotherapy in patients who had responded to platinum. And we saw, as we had seen in every study we've done, that these drugs have activity. These were all in patients who carried a BRCA mutation. And it was what most people were hoping to see is that it was at least as effective as chemotherapy. And what we saw is that if we looked at the patients who had not had a lot of prior treatment, so just 2 priors, there was what I would consider at least the same activity we would expect to see with our best platinum-based chemotherapy. And this was all in non-platinum-based chemotherapies. So now, if you think about the development field, we focused on the BRCA because that's where the biomarker was predictive of outcome. And now we've started to expand that. So now we're going into the BRCA-like state. And now we're going into all-comer populations. And at the next coming meetings, we're going to have 3 reports, probably 3, that are going to look at the use of it in the frontline setting. 1 will be in combination with chemotherapy. 1 will be in a BRCA-like state for maintenance. And one of them will be combining it with bevacizumab for maintenance. So 3 phase 3s. So we have this conversation in a couple months. We got even more data to tell you about.
And so what is the final, or what is the ongoing objective of these trials?
Right.
In terms
2.
of patients, just that are listening, or the caregivers that might be listening?
Right.
I mean, does it enhance the overall survival or the PFS, or what does it do to the patient?
So I think I can think of 4 kind of ways that this is going to go. So 1 is that we want to expand the population to patients that are not so narrowly defined.
So they will have more options now.
Correct.
The larger
that
population
more patients will have more options, right? So we know that, for instance, the state that we call HRD, which is not BRCA related, but just HRD, non-BRCA, that those patients seem to have some efficacy with these drugs. It's not as good as the BRCA patient population, but it's definitely something. There's a strong signal there. So these trials will be enhancing, it will be broadening the patient base that will be eligible for the drugs. The second is using the drugs in combinations to enhance their capabilities. The drug's capabilities, either one or both, either of them, so make them both better. And we have examples for that both of those have happened. We have this group that's mounting of patients who've had prior prior PARP exposure, and so now can we overcome the resistance that happened because those patients got a PARP and it stopped working, or it didn't work at all. Okay, so enhancing the current activity, overcoming resistance in patients who responded to it, and then the last category is essentially taking patients for which we would not expect the drugs to work and changing the tumor context so that now they do work. So overcoming resistance, enhancing index activity, and allowing these drugs to work in situations where they wouldn't work, and enhancing the base of patients who would be potentially amenable candidates.
That's very promising. Thank you for sharing that. And so, in terms of top 3 advances in ovarian cancer research in the past year, what would you share about the advances, and how do they translate into advancements for the patients in terms of their quality of life or their overall
progression-free survival, overall survival, quality of life, all of those features, financial toxicity, I'll add in there. So at least those 5 categories, all of them have had important advances this past year. I look at this more holistically, because for me, the discovery part of this is so important to continue to keep pushing the envelope. I know we've talked about this many times, but I feel like, we're not done. So we have a new class of drug that has a lot of activity. Obviously, PARP's important, immunotherapy is important, angiogenesis is important. Developing new drug targets that we can use more directed, targeted therapy, or targeted chemotherapy, expanding the opportunities for immune surveillance. All those things are available now, but none of them are, have led to the ultimate cure.
Mm-hmm.
So to me, the discovery piece is the thing that it's the carrot that keeps dangling in front of us, and we've got to keep pushing it. So.
Mm-hmm.
So discovery on the preclinical side there's lots of things going on. I think one of the most promising things that are being developed are ways to assess tumor microenvironment peripherally.
Mm-hmm.
So right now, our understanding of the tumor microenvironment, which is tumor cells, blood cells, lymphatic cells, immune cells, stromal cells, all those things that are in that area where the tumor is growing. The only way we can really understand that right now is to stick a needle in it, take a core out. And we can't do that as frequently as we would want to do to understand why the cancer is adapting.
Mm-hmm.
So adaptive changes. So we want to figure out a way to capture that in the blood so we can do it, daily if we wanted to. But, some way to come up with a functional assay that would tell us what's going on in the microenvironment so we can respond to it. Because right now, our strategy for treatment is, and every patient I'm sure who's watching knows this, is you get a treatment, you go through a couple cycles of therapy, and if you don't have a symptom, you'll get another CAT scan or a CA-125, and we'll say, hey, it's working or it's not working. And then we wait another 2 months and we do it that way. But if we knew, let's say after 2 weeks of exposure, that the drug was not going to work down the road, could we at that point change course or add something or drop something out? Could we do something there to change it? So can we be more responsive than just waiting to see what happens? And when it comes to things like PARP and immunotherapy and angiogenesis targeting, like using drugs like Avastin or bevacizumab, those are really important, and we could actually do something, right? Because we know what drugs actually combine well with some of these drugs. So those types of functional, peripherally ascertained, assessments of tumor biology, I think is the most promising thing that's being developed right now.
So tell me a little more about the blood test that you just mentioned.
So,
is it in the works or?
Well, there's lots of them in the works. None of them are validated yet. Obviously, they would be out in front of patients if we had them. But you'll notice that many patients ask, well, why are they taking so much blood during this clinical trial I'm on? And a lot of it's because of that. So what we're looking at are things like, fragments of tumor-associated DNA. We're particularly interested here, as I'm sure Dr. Sid has also mentioned on this program, that exosomes are really an interesting area because they're like little pieces of cell that are floating around. But we're looking for other factors, other chemicals in the bloodstream that might represent, for instance, the metabolism of the drug, of the tumor microenvironment. So all that stuff's being looked at, and it'll take time to sort through it, and that's why we need more and more participation on clinical trials. I know I make that plea every year or every month I come on this.
Which, interestingly, is my next question. That's a great segue, by the way. So tell us about the state of clinical trials that we are in right now, and why is it important for a patient to consider it, to sign up, and what message would you give to the survivor community, not just the survivors but the caregivers as well, who might be involved in the decision-making process on the importance of signing up for clinical trials for ovarian cancer? Yeah,
so I have this conversation every clinic, every single clinic, and I even had this with my own family. I had to go through the same kind of process. So as I mentioned before, we're not at the finish line. And so the only way that we continue to keep making these advances is we ask, hypotheses. And our job, my responsibility as an investigator, our responsibility is to come up with trial designs that lead to unambiguous results. Okay, the ambiguity is the killer. So patients want to know that if they're contributing, whether they get a benefit or not, that it's going to lead someplace, right? So our job is to come up with these ideas that make sense and to test them in a way that will actually get a result. So unfortunately, the participation on clinical trials nationwide is very low. And I know there's lots of reasons for why. Some of it is on our side. So we've been, in an effort to make a trial unambiguous, we sometimes narrow it so much that most patients can't get on a trial, right? Like the eligibility eligibility criteria are so strict that they can't get on. Number of lines of therapy, prior exposure to this drug, prior exposure to that drug, concomitant medications, other illnesses. So there is this balance that we're trying to make them broad enough so we can get more patients or offer it to more patients, but then but not make it so ambiguous that it could actually confound the results. So, but the other thing I get from patients a lot, particularly in patient families as well, and I have in my own family, was that most people look at clinical trials as, quote, guinea pig situations, experiments, like they're experimenting on me, and they all want to say, well, I don't think I'm quite ready for a clinical trial because I'm not sick enough. So both of those are just misconceptions that are really hard to overcome. So first, most of the trials that we do, even in phase 1, are done with either very strong rationale because it's coming right out of the preclinical space, or they're using these drugs in combination with drugs we already know work. So when we talk about doing a drug, let's say with a chemotherapy agent.
So just to intervene quickly. So in terms of, I think just from the patient perspective for a second, it could be that there is the risk factor of getting into a clinical trial that stops the patients from...
Risk in what way?
I mean, that's what I'm trying to investigate with you at this point. I mean, is it the risk factor, or what might happen if I sign up on the clinical trial? Is it going to work? Is it not going to work? I mean, what kind of assurance can the doctors give the patients so that there's something that is missing here? That is why the results are so low, right? In terms of the participation. So what could be done to make it higher for the patients?
Part of it's you.
Okay.
Part of it's you is making this kind of, is giving us a venue to basically present this so that people can understand what a clinical trial is. What I would say is that the standard of care options. So if I come in and I say, listen, I have this drug X, or you can get another chemotherapy, Taxol. So Taxol was drug X at one time.
True.
Okay, and it got to be where it is. But if Taxol was the end game, we wouldn't have to do any research. Okay, it's not the end game. So the assurance, there's two things I could say the assurance. One is that there's generally strong preclinical rationale or clinical rationale, like we already have data that says that this works. We're looking at a different setting. Or and so that's already that's your one assurance. The other assurance is that under a clinical trial, and I can't stress this enough, you get really good care because we are following a protocol that says if X, Y, if Z, then we go to A, and we have a safety margin built in. But in addition to that, you're also being evaluated very frequently. So if the drug, what a lot of patients feel like is that they get to this fork in the road and they say, well, if I go down this road, I made a mistake and there's no way to go back. But that rarely happens. Because if a drug's not going to work, just like with chemo, we're going to know in a couple cycles, and then we make a decision. And if it was Taxol versus drug X, and you went on drug X and it didn't work, Taxol's still available.
Right.
So it gets me to the other point I was going to get at, which was that when people wait too long. So one of the issues that come up with clinical trials is that we need patients that are healthy enough to get through enough exposure to know if it works. It's the other reason why we don't, give chemo as the last event, because we know that there's not going to be enough exposure for it to even work. So we want patients that fit eligibility criteria that are early enough in the game so that they can actually get a benefit if there is one there.
Right. Thank you for the explanation. And so with so many new and exciting advances happening, what does the present? Well, you talked a lot about the present, but the future of ovarian cancer look like, in your opinion? And what should the survivors and caregivers know and be aware of? Yeah. So one of the things I thanks for asking the question, because I think that what's the piece that's missing out of all this discussion is that despite the fact that the incidence rates and the mortality rates have basically been flat,
the number of women that actually have this disease is accelerating. And so the only way that can happen is that patients are living much, much longer than they have ever seen in history. I mean, we have pushed the envelope out so much farther now that that the ovarian cancer itself as an entity doesn't even qualify for orphan drug designation. So the prevalence rate is continuing to accelerate, and it's accelerating rapidly and has been since 2014, which incidentally was when the parts became available. So some of this is driven by that story. But what it's telling us is that as time goes on, because of this discovery work that we're doing with these clinical trials, these agents are becoming available to patients that wouldn't have had it today. So if we do, let's say we do another trial, we combine these drugs, it seems like it's efficacious, we get a new drug approval, the patient that's alive today who wouldn't have that option may have that option down the road, and when they do, they may get that benefit even further. And so the story of the value of the clinical effort, of the effort of making clinical trials available is in the prevalence data, and nobody sees it because it's not often talked about. So that's why I'm so excited to be able to talk about it here. But what I can say is that, and people who've heard me talk before will know this, but today it's actually now more than 10 times incidence. So the prevalence rate, the number of women who actually have this disease in the United States right now, is more than 10 times the incidence. More. It used to be, I used to say it's around 10 times. Now it's more than 10 times. So that's super exciting, because that's the value of having these agents, being able to develop them, getting them into patients, and making them live longer, and hopefully better. I mean, that's really the ultimate kind of balance to this. And so the good news about this is that with more available patients is that there's now greater interest in studying ovarian cancer patients, and many drug companies are coming back into the game. So that's, like, I was meeting, the reason I was late is I was meeting with another company trying to get another agent for us in this setting.
And so in the future, just in the next 5 to 10 years, I mean, we don't know beyond that, right? So in the next 5 to 10 years, what do you see the map of ovarian cancer looking like in terms of the patients living longer, their quality of life better, more clinical trial sign-ups? How do you envision the next few years?
Well, I would hope that more clinical trial sign-ups, because right now in the frontline setting we have like 5 phase 3 trials, so we are really moving fast. But just as fast as that's going to happen, the patients that have had who unfortunately develop a recurrence, they will have had exposure to these drugs. So our work right now is to anticipate that. So we already are starting with many programs now that are looking for patients who've had exposure to these other drugs, so that we can actually react to them and determine whether we can retreat or change it, so that we can actually use them again or use them in a different way. So the 5-year mark is going to be reflected on the current investigative environment. So I think what will happen is that frontline will change again. I think we're going to be getting into a space probably where we're using non-chemotherapy, so just biologicals. I think that will be on our plate.
That's a significant change.
It will be a significant change. And then also ultimately the incorporation of these 3 assets, so angiogenesis, PARP, and immunotherapy, I think those will kind of be better defined by that. 10 years, I don't know. But what I'm hoping for is that we'll actually have that assay developed that will allow us to actually make early changes. We'll have predictive biomarkers that will allow us to know what patients are going to do if they stay on drug versus not, and see if we can actually impact those responses. And we already have some signals about how this might work. Even looking at the SOLO-1 data, which was the big positive phase 3 trial that looked at the use of olaparib in patients who had a BRCA mutation, showed a lot of benefit in progression-free survival. But that benefit, again, is early. So the question is, can we interrupt that planned 2 years, give something else, and make it much different?
Yeah, thank you. And my last question, when it comes to our viewers and the survivors and the caregivers watching, what message would you have for our audience in terms of treatment and survivorship? Just a few words of your wisdom.
Yeah. Well, first of all, I want to thank them. I know certainly the ones that are part of our team are very invested and are very knowledgeable about the space. and continue to ask the hard questions for us. What are we doing to make it better? So I thank them for that, and I thank them for the ones that have participated in the clinical trials, because again, that's the only way we move the needle, and we have to move it. So but I do think that for I think at every decision point in the natural history of treatment for the patients that are on treatment, they should this idea of am I a candidate for a clinical trial? If we're going to change course, am I a candidate for a clinical trial? That should always kind of be there, and it's a completely appropriate question to ask their treating physicians, because I think you can get an honest answer one way or the other. Maybe they're not. That's fine, but. So the two things I would say is that one, ask that, and secondly, have faith in the standard of care. So lots of patients, keep looking for something that they think is better. Like, I'm holding back something I know is better, but if for instance they go on a randomized trial and they get randomized to the control arm, that's because it's the viable alternative, and it would be what you would get if you didn't go on the study. Right. So have faith in the standard of care. So push the envelope, have faith in standard care, and don't be afraid to ask questions about the trials.
Thank you so much, Dr. Coleman. Do we have any questions? Perfect. So thank you all for watching, and if you have any questions after you've watched, let us know. We'll get the questions to Dr. Coleman for you, and then we'll try our best to have him give explanations and clarifications as much as you require. And so thank you so much, and we'll see you again next time.
Thanks.